Hello, good morning and welcome to all of you. I am Rajesh Kumar Singhal and I am here to present a new lecture on the functions, transporters, uptake, deficiency and importance of the nitrogen. So friends, last 3 or 4 lectures we are discussing about the mineral nutrition. Okay. So in the first lecture we discussed about the basics of the mineral nutrition. Then we have discussed about the short distance transport of the nutrient. Then we have discussed about the long distance transport of the nutrient. Okay. We also discuss about the foliar uptake of the nutrient. Okay. So from today we will discuss about the function, transporters, uptake, deficiency and importance of the different nutrients. And today we will start with the nitrogen. Okay. So friends, you know that nitrogen is very important nutrient or mineral for the plant growth and development. Okay. And nitrogen is also a macronutrient. We already discussed that. What is macronutrients? Nutrient which require more than 100 ppm. Okay. So this nitrogen is a important element having the 1 to 5% weight of total dry matter. Okay. Second point is that this nitrogen it participates in the synthesis of proteins, nucleic acids, coenzymes, phytohormones and the secondary metabolites. Okay. So this nitrogen is important for in the synthesis of proteins, nucleic acids, coenzyme, phytohormones and secondary metabolites. So this compound is also crucial for the plant growth and development. Okay. Third point is that atmospheric nitrogen is only available for the plants which have nitrogen fixing bacteria. Example is the legume crops. Okay. Because legume crops have rhizobacteria. Okay. So only the plant which have nitrogen fixing bacteria are able to utilize the atmospheric nitrogen. Okay. Next point is that ammonium and amino acids are the dominating plant available nitrogen form in the acid forest soils. Okay. So friends, in general case, nitrate and the ammonium are the dominating form of nitrogen. What in case of acid forest soils, ammonium and amino acids are the dominating available nitrogen form. Okay. And uptake form of nitrate and ammonium already discussed. Next is the availability of nitrogen source depending on soil properties such as texture, pH, moisture, microbial activity. Like it is sandy, clay or loamy soils. Okay. So they have different texture. Also it is low pH or acidic pH or the alkaline pH. So nutrient ability also change with the pH. Also the soil in the drought condition or in the flooded condition or the microbial activity, availability of nitrogen fixing bacteria. Okay. So the ability of nitrogen depend upon so many factors. Okay. Next point is the there are two types of transporting system. That one is known as high affinity transport system. Second, LATS. Okay. So this high affinity are activated when the concentration is less than 0.5 millimolar. Or opposite of this, these low affinity transporters are activated when the nutrients are in more than 0.5 millimolar. Means when the nutrients are nutrient ability is higher, low affinity transports are activated. But when the concentration is less and requirement is high, then high affinity transporters are activated. Okay. Next slide is on the nitrate transporters. Okay. So friends, you know that there are number of transporters which are present in the plant for the uptake and the transport of the nitrate from soil to the fruits or the aerial plant organs. Okay. So this is a plant. Okay. So this NRT 1.1 and 1.2, they are crucial for the nitrate transporter in the roots. Okay. अगर हम बात करें तो रूट्स में कौन से होंगे? NRT 1.1 and the 1.2. Okay. Then this NRT 2.1, NRT 2.1 and 2.2, they are generally high affinity transporters also for the uptake of the nitrate in the root. Okay. Then NRT 1.5. Okay. This NRT 1.5 is for xylem loading. Okay. So this NRT 1.5 load the nitrogen into the xylem. Okay. Then NRT 1.8, NRT 1.8 used for the xylem unloading. Okay. So it releases the nutrient into the leaf. Okay. There are also one transporter that is known as CLCA that have important role in the vacuole storage in root and young leaves. Okay. So this CLCA is present in both root and the leaf tissue in the vacuoles. And this transporter is for the temporary storage of the nitrogen. Okay. Then NRT 1.7. So this is the phloem loading transporters. Okay. NRT 1.7. Then NRT 1.4. This is the petiole. This present in the petiole and it store the nitrogen in the petiole. Okay. 1.4. Then NRT 1.6. So this NRT 1.6 transport nutrient to the seed. Okay. Then last transporter is the NRT 2.7, which store the nutrient or nitrogen in the seed vacuoles. Okay friends, so this is about the nitrate transporters. So 1.1, 1.2, 2.1, 2.2. These are for the nitrogen uptake, 1.5 for the xylem loading, 1.8 for the unloading, 1.7 for the phloem loading, 1.4 for the petiole storage, 1.6 for the delivery to the seed, and NRT 2.7 for the vacuole storage in the seed. Okay. So these are the important nitrate transporters. Okay. तो आई थिंक याद हो जाएंगे, इतने डिफिकल्टी भी नहीं है. ओके. नेक्स्ट इज द अमोनियम ट्रांसपोर्टर्स. ओके. सो दे आर some ammonium transporters like AMT 1.1. AMT is the ammonia transporters 1.1, 1.2 and 1.3 for the uptake. Okay. And this 1.5 AMT 1.5 have minor role in the uptake, and there is a one specific transporters which is present in the pollen grain. This AMT 1.4 transports ammonium to the pollen grain. Okay. So they are the major transporters for the ammonium. Then there are some transporters which also transport the amino acids, and name is AIP1, AIP5 and LST1. These three transporters transport the amino acids. There are two transporters, like it is the Adopsis thaliana, DUR3 and the MIP, that is major intrinsic protein, that is the aquaporin family. They also transport the. Okay, so friend, this is about the nitrate, ammonium, amino acids and the urea transporters. Okay, now come to the nitrogen assimilation. So friends, you know that for the nitrogen assimilation there are two important enzymes, that is NR and the NI. This is nitrate reductase and second one is the nitrite reductase. Okay, so this nitrate reductase NR is a cytosolic enzyme and made up of three cofactors. Which one? F, AD, flavin, adenine, dinucleotides, heme, iron and the molybdenum. Okay, so it is made up of three components, while this nitrite reductase, it is made up of pyridoxine domain, binding domain, iron, sulfur, Fe, S cluster and the cyroheme. Okay, so this is about the structures, and now the reaction get regulated by these two enzymes. So this nitrate reductase converted nitrate to the nitrate, and this nitrate reductase converted this nitrate into the ammonia. Okay, very clear. So this is the reaction of nitrogen assimilation, where nitrate converted into the ammonium by the help of these two enzymes. Okay, this is known as nitrogen assimilation. Okay, but this process is very high. This process needs very high energy, and the energy requirement for the reduction of 1 moles of nitrate is equal to the 15 moles of ATP with an additional 5 moles ATP for ammonium assimilation. Okay, so friends, this is a cost. This is not a cost effective reaction. Okay, because there will be a lot of energy spent for the assimilation of nitrogen. So this slide is also very clear about the nitrogen assimilation, and this is also you heard about the GS GOGET pathway. So this is known as glutamate synthesis pathway. So friends, you know that there will be several reactions like photorespiration, nitrogen fixation, nitrate reduction, ammonium uptake. They converted into the NH2. Okay. Also, there are several enzymes like glutamine synthetase. This is known as GS, converted glutamate to the glutamine, glutamate to the glutamine. This is glutamine synthetase. Then this glutamine again converted into the glutamate, and this enzymes glutamate synthetase known as GOGET. So this pathway is also known as GS GOGET pathway. Okay, in which this compounds converted into the amino acids, and there are also several another enzymes like glutamate dehydrogenase, that is known as GDH. It converted ammonium into the glutamate, Then this glutamate again converted into the glutamine, glutamine to glutamate. Also, there is a enzyme that is known as AAT, aspartate aminotransferase. It converted glutamate and oxoglutarate to the aspartate, and again this glutamine is converted into the aspartate. So these are interconverted. Okay, so this is known as GS-Goget pathway for the assimilation of nitrogen, okay, into amino acids. So very clear, GS-Goget pathway. Okay. Now the form of the nitrogen in the plants. So there are generally two types of, that is assimilated form and the unassimilated form. So nitrate, ammonia and trace nitrate, these are the unassimilated form uptake by the plant, and finally assimilated into the proteins. Glycine and glutamate, already discussed about the GS-Goget pathway. Heme proteins in the cytochromes. Hormones, auxin, cytokinin, nucleic acid, base, FMN, NADP, and the alkaloids, polyamine like secondary metabolites. So this nitrate or ammonium converted into finally into the this product. So these are the two types of form are available in the plant. Okay. Next, what happens if there is a ammonium toxicity? What are the symptoms of the ammonium toxicity? So leaf chlorosis, stunted growth, and eventually necrosis leaves and the plant death. Okay, तो सबसे पहले आपका क्लोरोसिस होगा, स्टंटेड ग्रोथ होगा और नेक्रोसिस फाइनली हो जाएगा, उससे फाइनली एट द सीवियर कंडीशन देयर कुड बी ए प्लांट डेथ. आल्सो इट इनहिबिट द अपटेक ऑफ एसेंशियल कैटायन. ओके, क्योंकि आपका अमोनियम आयन है, दैट इज आल्सो ए कैटायन. अगर ये एक्सेस में होगा तो दूसरे कैटायंस के लिए ट्रांसपोर्ट्स को रिड्यूस करेगा. देन अमोनियम इंड्यूस्ड डिसऑर्डर इन द pH. ओके, इट रिड्यूस द pH. और pH में जब चेंजेस होगा तो न्यूट्रिएंट्स एबिलिटीज आल्सो चेंजेस. तो जो आपका आयन होम्योस्टेसिस है दैट इज आल्सो चेंज्ड. फोर्थ इज द एक्सेसिव कंजक्शन ऑफ शुगर फॉर द अमोनियम एसिमिलेशन कॉजिंग कार्बोहाइड्रेट लिमिटेशन. सिंपल सी बात है, अगर अमोनियम ज्यादा अपटेक हो रहा है, इफ देयर विल बी ए हाई अपटेक ऑफ अमोनियम, then there will be a lot of energy invested for the uptake of ammonium. Okay, जिससे क्या होगा, आपका कार्बोहाइड्रेट लिमिटेशन होगा. And next is the futile transmembrane cycling of the ammonium and the operation of energy intensive mechanism cause of ammonium toxicity. Okay, तो अमोनियम टॉक्सिसिटी से आपका मेटाबॉलिज्म भी इफेक्टिव होगा और जो एनर्जी इंटेंसिव जो मैकेनिज्म है दैट इज आल्सो इफेक्टेड. So these are the symptoms of ammonium toxicity in the plants. Then next point is the nitrogen deficiency. Okay, so it caused the stunted with the narrow leaves. Chlorosis caused by the nitrogen deficiency typically began in the older leaves and nitrogen is remobilized to the younger leaves. Canopy height is lower in grass or tillering as well as the number of seed per influence are also reduced. Okay, एक तो आपका कैनोपी हाइट रिड्यूस हो गया, टिलरिंग रिड्यूस हो गई, इसके अलावा नंबर ऑफ सीड पर इन्फ्लुएंस आल्सो रिड्यूस्ड. नेक्स्ट इज द देयर विल बी ए टेम्पररी नाइट्रोजन स्टार्वेशन. देयर इज ए डेफिशिएंसी ऑफ नाइट्रोजन व्हिच कॉज द रिडक्शन इन लीफ इलोंगेशन एंड लोअर शूट एंड रूट बायोमास. ओके, क्योंकि स्टार्टिंग में पढ़ा था 1 to 5% आपका बायोमास है दैट इज मेड अप ऑफ नाइट्रोजन. इफ देयर इज ए डेफिशिएंसी इन द नाइट्रोजन देयर विल बी ए रिडक्शन इन द बायोमास and continue nitrogen starvation. If there is a severe case of nitrogen starvation, then it cause the breakdown of nucleic acid and protein. Because already discussed that nucleic acid, proteins, and there are a lot of compounds made up of nitrogen. If there will be a less nitrogen or deficiency of nitrogen, these macromolecules start to be breakdown. Okay. Then next point is the mechanism to combat nitrogen deficiency. Then how we can improve the nitrogen efficiency or can reduce the nitrogen deficiency. So first point is the change in the root system architecture. Okay. Then second is the storage proteins, and third is the increased nitrogen use efficiency. So friends, you know that root is a very important for the uptake of most of the nutrient and also for the nitrogen. If there is a change in the root structure architecture, then there will be a change. So if we can improve the root structure architecture by improving the root length, root density, root biomass, root volume, or other root characters, it increase the nitrogen supply because it will improve the nitrogen transport. Okay, like lateral root elongations. Then storage proteins. Like there is a several proteins like vegetative storage proteins known as BFC. So this contain the 50% of total soluble proteins, seed storage proteins. So they can also provide the nitrogen during the nitrogen starvation. Okay, so nitrogen deficiency के time पे they can provide the nitrogen. Increase the nitrogen efficiency. This is very important point, and for this we need to improve the nitrogen uptake efficiency, nitrogen utilization efficiency, nitrogen translocation, nitrogen assimilation, and nitrogen redistribution. So when we can improve these traits, we can improve the nitrogen use efficiency. So friend, this is about the sort of the nitrogen. So there, you know that there will be a major uptake of form are the nitrate and the ammonium. So there will be some soil reactions like mineralization, emulization, nitrification, denitrification, ammonification. These reactions occur in the soils and increase the nitrogen ions. Then there will be some reactions like biological nitrogen fixation, addition of nitrogen fertilizer like urea, then addition of manures. It increase the artificially nitrogens. But there is some cases where this nitrogen can be lost, like runoff from the soil surface, nitrogen loss in the form of gas and volatilization process, also the leaching of nitrogen. So by this process, nitrogen can be lost. So this is the budget of the nitrogen, that several soil reactions, several artificial reactions, biological reactions, chemical reactions, and several losses determine the availability of the nitrogen. Then this nitrogen and nitrate and ammonia transport to the aerial portion by the several transporters. We already discussed about the NRT 1.1, 1.2, 1.3, 1.2.1. Okay, so then there will be enzymes like nitrate reductase and the nitrate reductase, which converted this into ammonia. Then there will be a GS-GOGAT pathway, which converted into the different amino acids in the proteins and the amino acids. Okay, so this is about the nitrogen budgeting. Then you know that if we can increase the nitrogen ability in the soils, how we can improve the nitrogen efficiency? So we need to increase the nitrogen availability in soil, and how we can improve the nitrogen availability by these reactions or by this reaction or by reducing this process. Then nitrogen acquisition by the roots. So by improving the root structure architecture traits, we can improve the acquisition efficiency. Then assimilation, we can improve by the this nitrate reductase and the GS-GOGAT pathway. So this can improve the nitrogen use efficiency. Then partitioning to the reproductive parts. If there will be an increased partitioning efficiency, the more nitrogen toward the reproductive part. Also the remobilization of nitrogen from the storage proteins and the senescence tissue also can improve the nitrogen efficiency. So friends, this is about the nitrogen. I hope you understand the today lecture. Thank you very much.